Welcome everyone to this September 2025 edition of the ARC seminar series. Today we have two talks, one from Duane Wesemann, Harvard University, and one from Joachim Clifford, University of Denmark. To briefly introduce Duane, he's an immunologist at Brigham and Women's Hospital and associate professor of medicine at Harvard Medical School. He's also an associate member of the Reagan Institute of MGH, MIT, and Harvard, the Broad Institute, and the Food Allergy Science Initiative. His clinical work focuses on patients with immune deficiency, immune dysregulation, and allergy, while his research work investigates fundamental principles of antibody diversification and adaptive immunity. His group has recently shown how somatic hypermutation influences the emergence of new antigen recognition, challenging long-standing models of antibody evolution. And I think this is also what he will talk about in a few minutes. And more broadly, his lab studies how immune memory develops and adapts across infection, vaccination, and allergy. Very much looking forward to your talk.
Fantastic. Thank you for inviting me to give this talk. And I'm going to just begin by sharing my screen here. All right, can you see that?
Perfect, yeah.
Okay, excellent. So I'll be talking about some research we've been doing to really push the antibody system to the limit with respect to somatic hypermutation, basically asking how far can somatic hypermutation take an antibody sequence with respect to antigen recognition. Okay, let's see here. All right, so this slide illustrates the diversity of antibodies. So there's what we refer to as primary diversity of antibodies, and this is um, the idea that antibodies, before you even see any kind of infection or vaccination, already have an extremely diverse repertoire of binding specificities. So this illustrates this idea here. For instance, there's like this yellow shape or this blue shape or this red shape or green shape you have on the left. These can illustrate different kinds of, vaccines or infections, viruses, bacteria, certain kinds of things that have different, shapes, basically. Um, so before you even see the yellow thing, you already have antibodies that recognize it. And this is due to an agnostic diversification process, which we'll introduce in a bit, but this generates a primary repertoire of diverse antibodies that can recognize this. So for instance, you already have antibodies that bind all of these things, whether the yellow, blue, red, or green thing. And so how this works in terms of an immune response is that when the yellow virus, so to speak, infects a person, the antibodies that recognize it expand. And then the cells that make the antibodies, the B cells that make the antibodies, can proliferate, and that's how you get a response to pretty much any kind of challenge that you can imagine. You already have antibodies that develop it due to this primary diversity. All right, so there's another kind of diversification that can happen to antibodies, and we'll call this secondary diversification, and this is basically referring to somatic hypermutation of antibody genes, which is tied to affinity maturation of the antibodies. So as an example here, you already have the yellow antibodies that bind to the yellow virus, and you can expand those cells that make the antibodies and therefore have a response against that due to primary diversity, and the secondary diversification process, the somatic hypermutation process, that basically takes the yellow antibodies and makes them yellower, basically. It makes the yellow antibodies able to bind to its target with higher affinity. And the same goes for the blue, red, and green as examples here. All right, so antibodies are comprised of four polypeptide chains. There's two identical heavy chains, two identical light chains, as illustrated here. Each of these chains has a variable region and a constant region. Um, and the variable region is named like that because that's the region that is extraordinarily diverse in its amino acid sequence, and that's how the antibodies can recognize many different things, is because the amino acid sequence can be very different in the variable region. And however, the constant region is like the name states, it's constant between antibodies. All right, so how the primary diversity takes place, how the repertoire, this primary diverse repertoire happens. It's through this process called VDJ recombination, and this requires an enzymes called RAG. There are RAG1 and RAG2 enzymes, and they are responsible for basically initiating this assembly process of the exons that encode for the variable regions of both heavy and light chain. So illustrated here are heavy chain on top in here, and then below is the light chain gene situation. So the variable region exon is comprised of three segments. They're called V, D, and J segments. And these segments are picked out from a variety of different similar kinds of segments, but differ slightly in their amino acid sequence that they encode for. And so assembly of different variations of these V's, D's, and J's creates this primary diversity of both heavy and light chain variable region exons. And then the constant region is encoded for by a different set of exons for the heavy chain and a different exon here for the light chain. All right, so that is primary diversity, RAG dependent and agnostic. It happens before the antigen is seen. However, the secondary diversity process, the affinity maturation process that happens through somatic hypermutation, this is a different thing. So this requires an enzyme called activation-induced cytidine deaminase, which, as its name implies, it deaminates cytidines, and that's how it initiates a mutation process. And this um, schematic here illustrates the different kinds of mutations that happen once you deaminate a cytidine. So it's a collaboration between AID, this cytidine deaminase, which deaminates the cytidine, turning into a uracil, and then various different kinds of DNA repair processes that can result in different kinds of mutations around this target site here. And so that's how somatic hypermutation happens. And typically after antigen encounter, where you then have proliferation and variation of the um, heavy and light chain genes to be able to figure out which ones bind better, basically. Um, and we're not going to talk about this today in this slide presentation, but just so we're all aware that the heavy chain constant region, that's where you get different isotypes of, say, IgM versus IgG, IgA, and IgE, that these can be swapped out within the heavy chain gene as well. But we're not going to talk about that today. All right. So how does affinity maturation happen? And so this schematic here I'll illustrate in somewhat simplified terms, basically the basics of how affinity maturation happens. So here is a B cell. The B cell expresses antibody on its surface, and here's just illustrating the heavy chain locus, making the heavy chain of the antibody. When a B cell encounters antigen, as well as other things that are going on, like engaging with T cells and immune responses happening, the antigen can be internalized and it's processed via MHC class 2 processing uh, stuff to be able to present peptides of this antigen on the surface of MHC class 2, which is on the surface of the B cell. And so what happens next is that T cells, which express a T cell receptor, T cell receptors can see peptide antigens on the surface of cells in the context of MHC class 2. And so this is how basically a B and T cell can coordinate together in response to an antigen. So the T cell is specific to the same antigen because it's, the piece of the antigen the B cell recognizes can be recognized by the T cell. And so in germinal centers, how this works is that there's fewer T cells than there are B cells. And so when the B cell is activated, it's getting signals from the T cell, it's getting some signals from this idea that it's engaging with an antigen, and this induces the somatic hypermutation process. So now you're getting variation of the antibody. You're getting different kinds of amino acid sequences resulting from this mutation process. And these B cells are dividing. So you get different versions of this B cell dividing. And so different versions of these B cells will have different versions of the antibody sequence and therefore might bind to this antigen differently by virtue of the fact of the different amino acid sequence. Some of them might bind better. And in these cases, there's more antigen that's internalized, and that translates to more peptide MHC complexes on the surface of the B cell. And because T cells are limiting, the T cells that are able to interact with the B cells are okay. So there's two principles here to keep in mind. One is T cells are limiting, and the second principle is that B cells in the germinal center are programmed pretty much to die without T cell help. And so that's how it works. If the B cells mutate such that there's a greater amount of antigen that's internalized, that will translate to greater peptide MHC density on the surface of B cells. And just by mathematics and probabilities, the T cells will interact more with the B cells that have more antigen on their surface. And the others that lose out to this competition will end up dying, and only those B cells that have sufficient antigen that's internalized to compete for this limiting T cell help will go on and survive. So the germinal centers have various different fates. They can keep going and keep dividing and keep mutating to have this iterative mutation and selection process to basically result in much higher affinity antibodies to the antigen. The germinal center B cells can exit to become memory B cells or differentiate to become plasma cells. And as I mentioned, the default state of germinal center B cells, I think it's generally believed to be that it's apoptosis. So the B cells are pretty much programmed to die unless they're rescued by T cell help. And so those antibodies that end up not being able to uptake as much antigen basically just lose out to the T cells that are, I'm sorry, the B cells that are taking it up. All right, so this next slide is the what my view is of what I think the current thinking is on how does somatic hypermutation get triggered in the first place for antibodies. So there are, I think, three main things that I think that need to happen. One is you need an antibody on the surface of the cell. So this generates what's called a tonic B cell receptor signal. And it's colored here in blue, which just indicates that the cell can tell whether there's an antibody on the surface or not. So that's number one. So this tonic blue radar signal is required. Then there's another signal that's, I think, thought to be required is that when an antigen is engaging the antibody on the surface, there is a different kind of signal, and whether it's really different in kind versus different in quantity of the tonic signal, I don't think anyone really knows. But there is some kind of a difference in understanding that the cell has, that now there's an engagement of the B cell receptor to some antigen. And that's what's colored here in this orange signal. That's meaning that there's some specific engagement of an antigen. And then the third signal is the T cell signal, which we talked about, and that's coloring here in purple. So when you have these three conditions take place, I think that's what the model is that I think that the field thinks that's required. So you need the receptor to engage an antigen. You need a T cell to be able to um, interact with the B cell in order to provide the signals and get the somatic hypermutation going and the selection process. All right, so that current thinking, the implication of that, I think it's fair to say, is the following. So, given this idea that a specific interaction of antigen and antibody is required in order to undergo the diversity process that is somatic hypermutation and affinity maturation. If that's required, then I think the implication is illustrated here. And I'm going to call this model the closed diversity model, um, just as a way to talk about these ideas. So the closed diversity model is as follows. So let's imagine that you take out all of your B cells and you line them up on this horizontal rectangle here. So you line up all those B cells and then you ask the question, how many of those B cells can recognize any given antigen? You pick a specific one, let's say ovalbumin. So I think it's fair to say that most of the B cells that you have in your body will not recognize ovalbumin. That is to say, they're not specific for ovalbumin. However, you will have a few that do just by chance because of the VDJ recombination diversity. You'll have some that interact and be able to bind to ovalbumin. So that's what's illustrated here, this reactive to antigen X, which is we're calling ovalbumin for an example. So you have some small portion that binds to this, and some people have measured this, like Mark Jenkins and so forth, has measured the number of B cells in a mouse that bind to a given antigen, and it turns out to be a very small fraction. Okay, so the closed diversity model would say that somatic hypermutation can operate on these antigen-specific cells in order to make them more avid, to increase the affinity. But the closed diversity model says that it wouldn't increase the breadth or it wouldn't increase what you can recognize. It just increases that you can recognize what you already recognize better, if that makes sense. So in other words, it ripens instead of expands the repertoire recognition thing. All right, so and this I think it's yeah, so there's some experimental observations which I think raise questions about the closed diversity model. And I'm just going to give a few examples here, but I think there's a lot of observations out there where people kind of, were head scratchers in terms of, well, maybe somatic hypermutation can increase uh, your antigen recognition capacity to other things. And there's examples of that. However, the response that that I've seen from the field is that you can't really prove that the antibody didn't really recognize it beforehand because of limitations of our assays ex vivo. So an example is this. So there's a paper, a great paper, Garnett Kelso's group, where they immunized mice with complex antigens, and they looked at the germinal center B cells that were responding to these immunizations, and they found that a bunch of them did not recognize the antigen, even though they looked like they were responding to the immunization. And so the interpretation here is what they came up with this term, this dark antigen. The interpretation was that the B cells are specifically engaging with some antigen. It's just that we don't know what it is. So it could be that our assay that we're testing ex vivo is insufficiently sensitive to detect the specific affinity that it has, but it's there. We can't see it, but it's there. Another idea of a dark antigen is that the antigen in vivo is somehow changed or modified in some way, where it's then in a form that's specifically engaging the B cell receptor. And so that's dark antigen, and that's fully in service to the closed diversity model because it's saying that, of course, there's a specific BCR recognizing the antigen, and that's why it's going. We just can't measure it. All right, and another idea out there, or other observations out there, is that there's a few cases, a few examples where research has looked at antibodies that are somatically mutated and binding to some specific antigen. But when you take the mutations away, in some cases, the reactivity seems to be lost. And so again, this could be interpreted as dark antigen that induced the antibody, or that the measurement, process that we're using ex vivo is just not sensitive enough, but of course it is specific. So those are some experimental observations that they've made to be consistent with the closed diversity model because of these explanations, which I think are is very valid explanations. I think it's true. I agree with the idea that the ex vivo measurement capabilities we have are insufficiently sensitive to really say for sure whether or not something doesn't bind to something. All right. That said, I think these observations are also consistent with another way to think about the role of somatic mutation and antigen recognition, and that's what we'll call the free diversity model, just as a way of talking to compare it to the closed diversity model. And I try to illustrate that here with this graph. And so in this graph, the y-axis here is specificity to a given antigen. So we'll call it antigen X again. And so the higher you are on this graph, the more specific affinity you have to that an antibody has that's to some antigen, to this antigen X. All right, so if this red line that cuts through here, that's representing here an x-axis here, that is the point at which we can no longer say that an antibody is specific to an antigen. And then, as we discussed before, I think it's fair to say that most B cells that are in your body are not specific for a particular antigen X. Okay, so illustrated here as well as this dark antigen. So that's this here, this non-detectable affinity. That's above the red line, so there's specific affinity, but it's just not detectable by us for whatever reason. And then there's this dashed line that's the detection limit, where we can say that there's some affinity that's happening that's specific to antigen X. All right, so the closed diversity model means that all of the action that happens through somatic hypermutation can only happen in this left quadrant here, where you already have specific affinity defined by the VDG repertoire. And nothing can happen down here with respect to expanding the recognition capacity beyond the primary repertoire. That's the closed diversity model. And as we discussed, that's where the dark antigen interpretation came from, because of course, there's specific affinity happening. All right, so I think these real world observations also are consistent with a free diversity model. That is that, there's sampling of the non-specific B cells to diversify them and to achieve what we'll call affinity birth by somatic hypermutation. And then once you have specific antigen recognition, then you get affinity maturation as per usual. And so this suggests that there's some kind of bystander B cell, like a true non-specific bystander B cell activation. And so the challenge here is, how do you really test this? Because if you can't measure a specific affinity, then you can never really conclude anything. But I think we thought it would be fun to test this because it's kind of an old question. This is Sir Macfarlane Burnett's publication in 1958, where he expounded on his clonal selection theory, which is a lovely theory that holds true. So clonal selection, that's what we illustrated, where the yellow antibodies expand to the yellow pathogen, and that's clonal selection. That's what happens. He mentioned in his book that this could be disproved by showing that the cells of a pure clone could, by appropriate manipulations, be induced to produce any one of a variety of antibodies. And so, we're not proving clonal selection theory wrong here, but it's just to illustrate this idea of, this question of, how far can you take a single pure clone, and how many different kinds of variety of antibodies can you make from that? Was kind of the fun question that he introduced a long time ago. And so that's the question we wanted to ask. And so this schematic here is illustrating again the observations that raise the question, really, where you immunize a mouse with an antigen A, and you look at the responding B cells, and most of the cells, or at least around half of the cells, don't detectably bind A. So this is consistent with a closed diversity model if you say that we just can't measure the affinity, but it's there. And then the other observation was where you take an antibody to antigen A, you remove the mutations, and then in some cases, there's no more detectable binding. All right. Now, how do you test this idea to see whether there's like this free diversity idea if you can't ever conclude anything about whether or not an antibody can really bind to something? So here's how we decided to go about doing that. So consider this experiment below here, where we take a mouse that only expresses a single pure clone. So let's call this an antibody against antigen B. And we determine by as best we can that it doesn't bind to X, Y, or Z. But of course, we can't conclude that it doesn't really bind in vivo, right? Because of dark antigen. All right, but so here's what we do. We take that mouse and we immunize it with antigen X, Y, or Z. So antigen X has nothing to do with antigen B or Y or Z, and antigen Y and Z are similarly very different than from everything else. And again, there's no evidence that they bind to anti-B antibody. Okay, so if we do this experiment and we find that some of the mice can bind to antigen X, for instance, then I think the closed diversity model can still be intact, because you can say that, well, this had, some binding to X we didn't know, and was kind of a lucky call because we just took antigen X off the shelf. Um, but if it ends up binding to all three of them, then I think, there's less probability that the closed diversity model can really be intact. So that's the idea. And so we started to do this kind of thing back a few years ago, where we took a monoclonal mouse that was a gift from Maria and Juan Lafay. So this mouse does not express RAG, so it doesn't make its own VDJ repertoire for T or B cells, but it does have a transgene for T cell receptor against ovalbumin, and it has a antibody heavy and light chain knock-in for an antibody that binds to hemagglutinin peptide, HA peptide. So we saw that the antibody against HA, it doesn't detectably bind to ova. It binds to the flow cell in SPR more than it does to ovalbumin. It binds to HA as we expected. We also took the cells from the mouse and asked whether or not, treating with complex HA or complex ovalbumin, changed things like calcium flux or antibody B cell receptor internalization into the cell, which is what happens when you engage the receptor on B cells. They internalize the receptor. So we measured these things by calcium flux and by B cell receptor internalization, and we didn't find any evidence that multimerized ovalbumin could engage the receptor in any way that we can measure. However, when we immunized the mice with ovalbumin, every mouse was able to undergo a germinal center phenotype B cell process where there's somatic mutation, and eventually they found solutions to bind to ovalbumin. So every mouse did this. And in addition to that, we put like haptens on the ovalbumin, and we found antibodies made to all the haptens that we tried, like fluorescein or TMP or biotin. We also saw that different parts of the ovalbumin were recognized. So different epitopes were recognized. So despite the fact that there's no evidence of binding beforehand, we found that affinity birth happened here. So there was some comments about this paper that, well, this was probably due to the fact that all the T cells recognize ovalbumin, and so this probably wouldn't happen in a mouse that had a polyclonal T cell repertoire. So we wanted to test that because we thought that was a fair assessment. So to do this, we made bone marrow chimeras where we took allylically marked B cells that were against the HA peptide, so the monoclonal B cells that are RAG deficient. They make an allele IgG-A, and they have a CD4 to 5.2 on the cell surface, and then other allylically marked wild type B cells. So we made chimeras. So remember that most of your B cells don't interact with any given specific antigen. So we wanted to model the overproduction or the overabundance of non-specific B cells by putting like 1,000 of the non-specific ones to one of the wild type ones, or 100 to 1 or 1 to 1. And so in the end, okay, so then we immunized with either ovalbumin or APC or CGG. And then we had these different ratios and these different colors. And so what we found is that we saw the wild type B cells responding in each case, as we would expect. However, we didn't see a serological response for the monoclonal antibodies. So we didn't see evidence of this affinity birth, at least in the serum. But when we took the cells out of these mice that were chronically immunized over many weeks with these different antigens, we saw that in each case, we saw that the non-specific B cells were involved in the germinal center. So they had a germinal center phenotype, up to, around 20% or so in the 1,000 to 1 chimera. But even in the 1 to 1 chimera, there were detectable at 0.1% level or so cells that were in the germinal center. And when we looked at the antibody sequences, we found that these cells were the B cells were mutating. So that's shown here on this graph below, where we're looking at the mutations along the heavy chain here, and the mutations tended to accumulate in the CDRs, so it looked like AID activity was happening, and there's quite a bit of mutation that was happening. So we were interested in this. So, are we not seeing the serological response because the B cells are incapable of diversifying that far to these antigens, or is it because the wild type B cells are just out competing the non-specific B cells, and so they're not given really a chance? So we repeated the experiments with just depleting the competing B cells. So we only have the monoclonal B cells together with polyclonal T cells by making a chimera, 1-to-1 chimera with mu-MT mice and the RAG deficient B cells that are HA positive. We also did a completely different antibody starting point. These are the B18383 mice. So it's a B18 heavy chain and a 383 light chain. Kind of a complicated name just to say that this antibody is innocuous. It's just a different kind of an antibody starting point, where it's pretty purely monoclonal, despite the fact that it's not RAG deficient. This B cell receptor is innocuous enough where greater than 95% of the B cells are this specific antibody. All right, so and just to briefly show that we did the analyses and the calcium flux and the B cell receptor internalization from a large panel of antigens, and we didn't have any evidence that these antibodies were polyreactive, and they didn't bind to any of these antigens that we immunized them with. So in this case, when it turns out that we immunized the that the mice were able to respond to all the antigens serologically when we removed the B cell competition. So again, we immunized with ova, albumin, APC or CGG, and this on the left here, these are the B cells that initially recognize HA. In a polyclonal T cell environment, all the mice responded to all the antigens. In particular, CGG, it looks like they responded almost as quickly as they did as the wild type mice, which was kind of surprising. Ovalbumin, it took many weeks for the serological signal to arise. sorted out B cells from the mice that were antigen specific, and we cloned several of the antibodies and found that several different epitopes were targeted. So this graph below is just competition analysis of different monoclonals that were cloned from the mice immunized with the different antigens as indicated here on top. The graphs on the right show the B18383 mice, so similar to the HA chimeras, the B18383 mice, all the mice responded to all the antigens, and we took some monoclonals from the HA mice and competed it against some sera from the B18383 mice to illustrate that the mice were making antibodies to different epitopes on the antigens as well in that case. So we thought this was pretty cool, and I was expecting that these antibodies would have like crazy amount of mutation, but it turns out that the amount of mutation that these affinity-born antibodies had were basically similar to what we find in a regular germinal center. So because of time, I think I'm just going to skip to the bottom graph here, just to illustrate this. These are logograms that show the kinds of mutations that were happening in the different conditions. So the top condition here on the left, this is the one-to-one HA wild type chimera, where we didn't think that selection was happening. And so these mutations, I think, illustrate just the intrinsic mutability, for the most part, of the IGH locus. And here with ovalbumin, APC, and CGG, these are the heavy chain sequences that were from antibodies that were specific against these different antigens. And what we thought was really interesting is you see very similar patterns, like this T is, pretty dominant in everything, despite the fact they bind to different things. You see this M in everybody and this F in everybody and so forth. The B18 heavy chain, we see the same thing. And on the top passenger, this is a publication where there's stop codons in front of the B18 heavy chain, but it comes from B cells that were undergoing, some immune response, and the passenger was receiving mutations but was not expressed. So these are non-selected mutations. And then here's the OVA, APC, and CGG reactive data sets, and you see similarly that you get very similar patterns of mutations. So this graph illustrates this in a different way. So up on top, you see the sequence, amino acid sequence of the antibody for HA, heavy chain, and then on the right, B18. And then the y-axis is all the 20 different amino acids of, so basically where you see the circles, that's where you're getting a mutation to a specific amino acid. And if you see the color red, that means that mutation outcome, that specific outcome, is present in all the sets. And I think that means it's only present in that single data set. For instance, this pink one here in the B18 OVA, we only find that mutation in this anti-OVA data set. And then the other colors are other combinations of sharedness between the data sets. And so what you see here is that like nearly all the mutations are shared, and there's quite an abundance of the shared with everybody. So this is pretty cool because you're getting, quite diverse recognition, but you're getting a lot of the same mutations, and whether or not they're involved in the recognition or not, they're at least tolerated and they're carried along in a big way. Okay, so another cool thing that we thought of, this experiment sort of taught us a little bit, is when we looked at the different mice. So each mouse here is a different color, and we made evolutionary trees of the antibody sequences. And in some cases, like the albumin case, it looks like the mice had a very distinct clade of mutational evolutionary trajectories to find a solution to bind to ova, whereas different mice had completely different ways to do that. And this kind of, I think, is a fun reminiscence of this idea of frozen axicants from François Jacob, who was thinking about, what if you just replayed evolution? Would you get the same thing, or would you get completely different potential outcomes? And this just illustrates that you can start with the same starting point and ask evolution to find a solution to some selection event, and you can find completely different paths to find that. So I think in the end, this illustrates is that a free diversity model way of thinking could be valid, where you have this idea of non-specific B cells that can undergo somatic hypermutation and affinity maturation, affinity birth and maturation. I think the implications are pretty cool because number one, that means that the competition process that tends to control the affinity maturation process in germinal centers isn't just for the specific B cells, that the competition rule is the rule for the entire set of repertoire. So, in other words, we see the specific antibodies respond most often because they're most fit for the competition process, not because specificity is required to initiate the process, if that makes sense. The other implication is that you can have recognition specificities go beyond the VDJ repertoire, which I think is a fun thing to think about. How this happens, we're not quite sure, but I think what we'd like to propose is that non-specific antigen uptake into B cells is sufficient, or rather, I should say this, the amount of peptide MHC complexes that results from nonspecific uptake is sufficient for a T cell to engage a process to get the B cell going to somatically mutate. So in other words, if a T cell arrives at a location and finds no cognate B cell, it can always make one by asking evolution to start the process because B cells, their APC function is still intact, despite the fact that they may not recognize what their B cell receptor. Their uptake capability is still intact, even though a very small amount is sufficient to get the T cell to say, hey, how about you mutate? I don't know who you are or what you bind, but how about you start to evolve? And then you'll get a process that you can get this competition. All right, so due to time, I think I'm going to skip this part. I'll just summarize it by saying we think that specific T cell recognition is required for this process. And then the T cells can then just ask the B cell to evolve until it finds a solution. And so I think this is a fun illustration of this more ancient role of antibody diversification. So, cytidine deamination is more ancient than a RAG-dependent diversification process for antibodies. You see AID-mediated diversity for diversity's sake happen in certain circumstances in different animals across phylogeny. Whereas in humans and mice, for instance, it seems like the AID process is more focused on, you know, ripening the VDJ recognition profile. But I think we can realize that perhaps the AID system remembers its origins as a diversifier. So, in conclusion here, to summarize, nonspecific and bystander B cells can obtain a GC phenotype and diversify in a polyclonal T cell environment in the presence of competing B cells. So they diversify. Intrinsic IG sequence SHM-mediated mutability is sufficient for a wide range of new affinity acquisition. So we haven't found the limit. Everything we throw at the mouse, the antibody can find a solution using run-of-the-mill pedestrian mutation profiles in intrinsic mutable locations. It can easily find, it seems, it can easily find a solution. So why then do we need a primary repertoire? And so what I think this might provide some insights into is that the primary repertoire may not be required to find a solution to antigen recognition, but it does provide a solution to the timing of when that can happen. So in the wild type repertoire situation, you have fast and consistent responses in all cases, even though it may not be required to recognize. In some cases, when SHM is the somatic hypermutation is the only thing available, then it can take sometimes up to like 10 weeks to find a solution, which is kind of interesting. All right, and so then we talked about this idea of clonal competition. It regulates things all the way down to zero. So the most important part here is to explain that Teng Zhou and Avanish Gautam are the postdocs in the lab that have done most of this work that I presented. Shahab Sakai and Gaspar Pacheco and Vishal Rao did a lot of other work as well that was really important to get the data out. Um, and with that, I will close and take any questions.
Super nice talk. I mean, to me, this really seems like we have to change a bit of the textbooks that we um, have. But I have many questions. So maybe one of the most basic questions, but I think you have answered this. So we can be absolutely sure that the mice are not leaky or anything, right? So we can be absolutely okay. Yeah.
Yeah,
Um,
that was when we cloned the antibodies, we're able to see
yeah.
the sequence came from
Yeah.
the original.
Yeah. Yeah. And when you show these trees in one of the last slides, so the parent sequence is really then only has only affinity to the target of the mouse, and then they mutate, and then they slowly get affinity to the antigen that you give to the mice. Is that yeah, yeah. And then have you measured of the expressed antibodies or the cloned antibodies? What is their affinity? So do they really get high affinity with SPR or something like that? Yeah.
Yeah, in some cases they do, and it's
Yeah.
in it for the so Avnish and Sachin and others in the lab, they measured the affinity to these antibodies. In some cases, they go up to minus 10,
Oh my.
minus 9, I think.
Yeah,
Might be a couple cases of minus 12. Avnish, you can correct me if I'm wrong.
that is
If
crazy. Yeah.
there. But it tends to be associated with kind of antigens. So the ovalbumin, for whatever reason, tended to make lowish affinity, like
Mm.
-8, -7. Whereas I believe the APC and CGG, they're able to have examples of really high affinity, which was interesting.
That's very interesting. And then, I mean, I asked this before already to you, but maybe you have an answer now. You don't have structures yet, how to see how the antibody goes from being specific to this antigen to then
Mm.
the other antigen. And
Yeah,
then
that's yeah.
do the anti so do the mutated antigens that gain the new specificity, do they retain specificity to the original antigen? Are they
So
fully
yeah,
dual reactive or yeah?
the second question, so Avanish and Gaspar in the lab, they've checked to a bunch of different I'm sorry, they checked to the original antigen,
Yeah.
and Gaspar found, I think, one or two examples where the HA specific B cells that evolved to bind as other things retained the HA binding. Whereas in, I think, greater than 90% of the time, they completely lose binding to the original antigen. In terms of the structure, I think it's a great idea, and we would love to do that. And we should probably reach out. I think you
Andrew
suggested.
Ward or something. Yeah, yeah, yeah.
Yeah, I think that would be really cool because, to kind of define what are the principles of like this affinity birth are there like common features
Yeah.
there?
And then last question before Gua, I think, had a question. And do do the different mice, because you showed those different mice, do they find all the same epitope, or do they bind, or do their antibodies bind different epitopes?
Love the question. Avanish is looking at that as we speak. So
Okay.
uh, there are examples where there's common epitopes being recognized
Mm.
by both antibody starting points.
Mm.
And I think Avnish just found an example of a unique epitope, though, from one of the starting points. So it's probably a mix. I think, it raised the question in my mind, are there certain epitopic features that tend to be prone to recruit this kind of affinity birth process? Because we have completely different antibody starting points recognizing the same epitope on a complex antigen.
Yeah,
Um,
yeah, and then yeah, just one more question, then Gua. Do you find the same immunodominant epitopes as if you were to give the antigen to a normal mouse? Like, do you converge epitope rise to as if you had given it to a normal mouse? Yeah.
You're asking all the great
Okay.
questions.
Okay.
So Natalie in our lab is doing that for like HIV.
Yeah.
because that's been mapped out really carefully in terms of what are the immunodominant epitopes and we're I don't know if we're doing it right now, but it's on the thing to do is to do all this in wild type mice to
Great.
compare
Yeah.
because
Yeah.
you're exactly right.
Yeah.
That's a great question.
Great. Gua, you had. Sorry for keeping you. Yeah.
No, no, that's great. Great talk. I really enjoyed it. I have two questions, but maybe one just follow up on what you just mentioned. This is kind of related to this dark antigen hypothesis, maybe. I mean, because I don't think that the free diversity really addresses it, right? I mean, if you have like some new clone that comes and you don't know what this is specific to, maybe it's another epitope, or maybe, I mean, how does it fit in your framework?
Yeah, so I tried to illustrate where it fits in that graph that I showed in one of the last slides. So I think dark antigen is a thing. I think it's a great concept. It's a great way to think about it because, we can't measure all the things. So in an individual case, I think it's still that we don't know. If you take an individual case and say, did this arise by affinity birth or did it arise because of some specific interaction, but we don't know. I still think we're in this area where we can't tell on an individual case. But if we look prospectively, and that's what this experimental system was set to do, is we
Mm.
take beforehand where we define a set of antigens where we take a panel. So it's winning by numbers basically. So, and what we found was that in each case, in two separate starting points, all the antigens were able to find recognition. So there, I think it's just, is it absolute proof? No, it's not absolute proof, but I think the more antigens that we find this, the less we can ascribe it to dark antigen. That those instances in that experimental system. But I think you're right. If we find an antibody out in the wild and find that it didn't, it loses binding, then we don't know because I think I think what this research shows is that dark antigen and affinity birth stand on equal footing as considerations in those cases. Does that
Yeah,
make sense?
yeah, very much. Now to my real question. So it seems like you have some kind of repertoire scale competition. Essentially, that's what you're saying for the clonal expansion. Now, my question is, do you think it happens at the germinal center level, or different germinal centers will start with different cells? So, in a more of a real life situation, is the
Yeah.
competition for newcomers is at the germinal center level, or germinal center, it kind of blocks if it
Mm.
has sufficient affinity, and you can only seed new ones with
Yeah,
fresh
it's a
data.
good question. I think Gabriel Victoria is the best person to ask that question to. I think his recent bioarchive paper, I think, which is beautiful. It's really looking at the affinity spectrum in these different germinal centers, and I have to look at it again carefully, to give it justice. But I think, and anyone correct me if I'm wrong here, but I think the idea that one of the ideas that came out of that was it's kind of surprising how diverse the affinities can be. So, the winning clones, it would be, kind of expected that you always see the highest affinity win out in the clonal bursts in all the germinal centers. But I think what Gabriel was finding is that you get low affinity bursts as well. And you have this, so I just remember thinking when I was learning about this data is, it looks like diversity for diversity's sake is one of the outputs of a germinal center because you don't always have the winning clones win. On average, you're getting affinity maturation, but through the process, you're getting kind of these random bursts of even lower affinity things. And then you have this clonal replacement that happens over time. So I think it's a dynamic process, and I think it's very complicated. But in the end, I think in addition to what Gabriel is showing in those models there in what looks like a very physiologic setting. Our, push it to the edge contrived system, I think, shows that the B cells are at least capable, even if they don't even have any specific affinity that we know of, that there is a capability for them to be involved somehow. So it might be one of these cases where, you have abundant epitope masking in a very chronic antigen where you don't have your repertoire. Your repertoire has failed you because you have a chronic antigen for months and months and months. All the epitopes are masked. The system won't die. As long as you're still alive and you still have cells circulating, any T cell that's still specific for the antigen can make a new B cell against a new antigen that's not masked. So I think that's kind of the scenario. How exactly that happens, I think more research needs to be done.
Thank you.
Super interesting. You mentioned in one of your slides polyreactivity also. Maybe I just ask the question, for the antibodies that you cloned and then tested for binding, are they polyreactive? Also more with respect to the question, because you mentioned that across antigens, some of them find the same mutations and so on. So then maybe I would be thinking maybe, yeah, are you just converging to sticky clones that can kind of bind everything?
Yeah,
Yeah.
great question. And
Yeah.
Avnish is actually testing the specific antibodies that have come out of the affinity birth system,
Mm, mm,
checking to see how specific they are. And I think in all cases so far that they're not polyreactive as far as we could tell.
mm,
But Avnish, you can correct me if I'm wrong, but that's kind of an ongoing experiment that we're doing. So we don't have the full data set in yet, but we're not finding a lot of polyreactivity.
mm, mm, mm. Yeah, I think we have to move on in the interest of time, but yeah, unbelievable. I think there's so much more to be done there. Yeah.
Thanks
Uh,
so much.
thanks so much.